Hey guys, Jason Matthew here from Trinidad and Tobago, and welcome to Biochem JM, the YouTube channel. Today we're going to be doing a glycolysis MCQ worksheet. So have your pen and paper ready guys, and let's get started. First question. In eukaryotes, glycolysis typically occurs in the, and you have 5 choices, one of them is correct. So you write down the letter for the one that you think is the best answer for this question. So again, in eukaryotes, glycolysis typically occurs in the, and if you had B, the cytosol, then you are absolutely correct. Alright? If you had mitochondrion, but then you have no idea what's going on here. Alright? Because we all know that glycolysis is for anaerobic respiration. It doesn't involve oxygen, it doesn't involve mitochondria. Alright, so again, in eukaryotes, glycolysis typically occurs in the cytosol. Next question. Which of the following enzymes catalyzes the transfer of a phosphoryl group from ATP to glucose? Alright, so again, which of the following enzymes catalyzes the transfer of a phosphoryl group from ATP to glucose? So you have 5 choices there. Which one is the correct one? So whether or not you knew anything about glycolysis, if you know your basic enzymology, which have been taught in year 1, you should know the answer for this question without even knowing anything about glycolysis. Alright, because if you are transferring a phosphoryl group from ATP, alright, to glucose, let's break down that. You are transferring phosphoryl group ATP glucose. Now if you're transferring, that's a transferase enzyme. So it belongs to the transferase class of enzymes. Alright, and what type of transferase enzymes would use would transfer a phosphoryl group from ATP? That's correct. That those subclass now will be kinases. Alright, so the major class is transferases and the subclass will be kinase enzymes. And I mean, down here now you only have one kinase enzyme, so you already know what the answer is, but let's continue. And the next clue here is glucose, which is a hexo sugar. So therefore the answer is, and drum roll, the answer is D, hexokinase. And I just showed you all how you could work it out. And if you're not sure about the answer, there are ways in which you could figure out stuff. You could definitely, in a multiple choice, you could rule out 1 or 2 answers being absurd, and then you start to break it down. So let's continue. The anaerobic conversion of 1 molecule of glucose to 2 molecules of lactate by fermentation is accompanied by a net gain of, so the key thing here guys is a net gain, and you're converting 1 molecule of glucose to 2 molecules of lactate. Alright, so what do you think the answer is? You have 5 choices. One of them is correct. That's right, 2 ATP. Now the whole idea here is net gain. Now somebody might have been tempted to say 2 moles of NADH. Alright, but the thing here is that when you go from glucose to pyruvate, you really do make 2 moles of NADH. But then when you go from the 2 molecules of pyruvate to 2 molecules of lactate, those 2 moles of NADH is now reconverted back to 2 molecules of NAD+. So there's no net gain of NADH. The 2 NAD+ that was converted to 2 NADH, those 2 NADH molecules now, when you're going from pyruvate to lactate, those 2 NADH molecules would have been converted back to 2 NAD+. Alright? So the true net gain here is 2 ATP. Alright? So remember that. When you go from 1 glucose molecule to 2 molecules of lactate, the net gain of ATP is 2. The conversion of 1 molecule fructose 1,6 bisphosphate to 2 molecules of pyruvate by the glycolytic pathway results in a net formation of that's right, the answer is 2 NADH and 4 ATP. Now that might be a little trickier, because instinctively you would have wanted to say probably D, which was 2 NADH and 2 ATP, but that's wrong. Because you see, you have to be very careful. If you were starting from glucose, alright, if you were starting from glucose and you were going to 2 molecules of pyruvate, well then fine. Then the answer would have been D. But look at where you are starting. You are starting from fructose 1,6-bisphosphate. It means that you have missed, alright, the 2 priming actions that would have consumed ATP. Alright, so you have gone straight into the payoff phase. You're going to get all of the ATP as a net gain. So that's why you have 4 ATP. Alright, so again, if you're still not getting what I'm saying, pull out your glycolytic pathway, because I always think that you should have one of those printed out somewhere. Pull up your glycolytic pathway and look where fructose 1,6-bisphosphate is, and go down from fructose 1,6-bisphosphate until you reach pyruvate, and you will see that from fructose 1,6-bisphosphate to pyruvate there's no consumption of ATP, but there are 2 steps where 2 ATP each is produced, so you get a sum or a net gain of 4 ATP. So check it out. During glycolysis, the steps between glucose and the formation of the triose phosphates, and you have to finish that sentence. So during glycolysis, the steps between glucose and formation of the triose phosphates, which one there will best complete the sentence? So give it a little thinking about it. Some of these answers looking kind of close, it's sounding a little tricky, so give it some time and check it out. Well, first of all, ask yourself, what is a triose phosphate? Well, it's just simply, triose mean tri, 3, so there's a 3 carbon sugar with a phosphate group. So when do you get 2, well, I kind of given the answer there, but when do you get 3 carbon phosphates? When do they initially form? That's in the aldolase reaction, where you split them, right? Where you split the 6 carbon sugar into 2 3 carbon sugars. So they're saying the steps between glucose and that, the formation there. What is the best sentence there to complete that sentence? Well, I will go with consume 2 ATP molecules, because when you go from glucose to the formation of the triose phosphate, which is the aldolase reaction, those steps involve the energy investment phase, and during the reactions of the energy investment phase, that's where you consume 2 ATP molecules. Alright, so in skeletal muscles, the NADH that is produced by glycolysis under anaerobic conditions, in other words vigorous exercises, not enough oxygen is reaching the muscle, the NADH is regenerated to NAD plus by the conversion of, and I like this question because it has a little, the 2 possible answers are very close to each other. Alright, so you should have probably limited 1 or 2, and you could be trying now to decide between, is it C, where you're going from pyruvate to lactate, or is it E, where you're going from acetaldehyde to ethanol? Those are 2 close ones. Now, why I would say that 2 of them are close answers is because here's the thing. When you go from pyruvate to lactate, or if you go from acetaldehyde to ethanol, in both cases, NADH will be converted to NAD+. In other words, one of the criteria in the above stem here is fulfilled. In other words, NAD plus is regenerated. But here's the thing, read, and that's why you must in the exam read your questions very carefully, because this question is saying in skeletal muscle cells, and because it specifically says skeletal muscle cells, there can only be one right answer, and that answer is pyruvate going to lactate. The answer is C. All Alright, so let's go on. Glycolysis in the erythrocyte produces pyruvate that is further metabolized to. But first of all, what is an erythrocyte? Well, you should have correctly said that an erythrocyte is a mature red blood cell. So you all know the deal about the red blood cell and the irony of its own life journey, right? The red blood cell contains the pigment that carries oxygen, which is hemoglobin. Now, for the red blood cell to maximize the amount of oxygen it can carry, it wants to hold the maximum amount of hemoglobin. So what it does is that it's pretty empty on the inside. It just contains hemoglobin. And when I say empty, it means that it's lacking organelles. And one in particular for this topic is it's lacking mitochondria. Therefore, the pyruvate cannot enter the TCA cycle because there's simply no mitochondria there. There's no electron transport chain. So the pyruvate is then converted to something else, and it's converted to lactate. All right, so I hope you got that, guys. You know that's a classic biochemistry thing, as you should have that. All right, so let's go to the next question. Which of these cofactors participates directly in most of the oxidation-reduction reactions in the fermentation of glucose to lactate? And you see that word fermentation. You know, when we think of fermentation, the layman might think of alcohol production. But also note that lactate production is also a form of fermentation. So we're asking there which cofactors that typically participate in the oxidation-reduction reactions in the fermentation process of converting glucose to lactate. And the answer is, of course, NAD plus NADH. The steps of glycolysis between glyceraldehyde 3-phosphate and 3-phosphoglycerate involve all of the following except, right? So it means that 4 of them are correct. I shouldn't say that. Let me rephrase that. 4 of these options occur between glyceraldehyde 3-phosphate and 3-phosphoglycerate, but 1 of them don't. So which one is it? So the answer here is oxidation of NADH to NAD+. And that should have been a given without even reading the question. Because you know in glycolysis, NAD+ is converted to NADH. The regeneration of NAD+ comes later on in life. Alright, so during the actual steps of glycolysis, NAD+ is converted to NADH and not the other way around. Alright, so that's the wrong one. In which of the following metabolic conversions is ATP consumed during glycolysis? So look at these reactions here, guys, and tell me which reaction you think there will consume ATP. Now, it's much easier than you might think, because a lot of people, when they see chemical equations, they start to get, a little nervous. I see them start to fidgeting and, school sweating and all kind of thing. Don't. It's a very easy question. Despite all those big words there and all those arrows and so on, it's a very simple question. So look at those reactions and tell me which one you think will use phosphate. Alright, so you should have come up with C. Look at it. What's the difference here? You have a substrate, which is glucose, and the product is glucose 6-phosphate. Alright, the difference here between the two is the phosphate group. Where did the phosphate group come from? You consumed ATP. You took the phosphate from ATP. Now, this question is asked, but let me ask you all now, what do you think is the enzyme that catalyzes the reaction of glucose going to glucose 6-phosphate? So you should not have hesitated, because this should be coming like a reflex action now. Glucose going to glucose 6-phosphate in glycolysis is catalyzed by hexokinase. Excellent. The process by which ATP is formed from ADP in glycolysis is referred to as, well, I think the 2 possible answers you should be deliberating between will be substrate level phosphorylation and oxidative phosphorylation. Those are the 2 ways in which the cell can form ATP, but it's an MCQ. There can be only one right answer. And the thing here is that ATP is formed from ADP in glycolysis by the process called substrate level phosphorylation. Alright, and I will always tell you all, go and read up more about substrate level phosphorylation. You can check my YouTube videos, they have some information there as well. Alright, so the next question here now is, which enzyme is involved in substrate level phosphorylation and glycolysis? So think about it, you know. Now there are actually two enzymes that are involved in substrate level phosphorylation and glycolysis. But looking at these options here, there's only one possible answer you could get. That answer is pyruvate kinase. But you should also know the other enzyme that also does a substrate level phosphorylation step. Because there are two substrate level phosphorylations that occur in the glycolysis. Two of the enzymes will catalyze those reactions. One of them is pyruvate kinase. But I'm just saying, go back to your glycolytic sheet if you can't remember, look at the glycolytic pathway and see which other enzyme will also catalyze a substrate level phosphorylation. Alright, it's very important because in your exam, instead of pyruvate kinase, you could have the other one, which I'm not going to let you know. Look at the glycolytic pathway if you can't remember and see which one it is. Which of the following metabolic conversions is considered to be the major control point of glycolysis? So which of the following metabolic conversions is considered to be the major control point of glycolysis? Well, there are a few things that you need to know to get to that answer. First of all, another common question is this: what is the most regulated enzyme in glycolysis? And that's a question you might more see, so you might just immediately say the right answer, which is PFK1, phosphofructokinase 1. So basically what this question is asking you all to do is to give that reaction that PFK1 catalyzes. So what is the reaction that PFK1 catalyzes? And that's correct. When you go from fructose 6-phosphate to fructose 1,6-bisphosphate. So excellent job. Alright guys, so the last question for the road. Alright, so if you have been getting all correct so far, this is the last one. In the alcoholic fermentation of glucose by yeast, thiamine pyrophosphate, which is abbreviated as TPP, is a coenzyme required by, there's one of these guys here. Well, obviously, you know, if you know the enzymes that are required for alcoholic fermentation, well then it's easier for you all. Alright? Because you can rule out some of these immediately. Like for instance, anybody who says lactate dehydrogenase is because they have been drinking probably too much. Alright? That's out there. I mean, it's alcoholic fermentation. Lactate dehydrogenase is involved in lactate fermentation. So if anybody who says C, you need to go into the biochem hall of shame. Alright? So C is out. We already know hexokinase by now, common people. You all should know what hexokinase, what reaction that catalyzes is the first step in glycolysis, which is glucose going to glucose 6-phosphate. So then you're down to 3. Now, curiously enough, trans-aldolase is kind of close, because actually trans-ketolase, that enzyme uses TPP, but not trans-aldolase. But anyway, trans-aldolase is not involved in the alcoholic fermentation of glucose by yeast. So you're down to the two enzymes that are involved in alcoholic fermentation of glucose. You have alcohol dehydrogenase and you have pyruvate decarboxylase, pyruvate decarboxylase, alcohol dehydrogenase. Which one will you choose? One is right, one is wrong. And the answer is pyruvate decarboxylase. Alright, and that is the last and final one. So guys, as usual, I will ask you all to LSC, which is to like the video if it did something for you. Subscribe to the YouTube channel, become part of the Biochem Nation. Alright, and finally, comment. Let us know if these worksheets are helping you all, if it helped you in an exam, it helped you understand glycolysis some more. Give us some feedback. We really appreciate it. And as usual, thank you for the love and support that you all send our way. It means a lot and it keeps us going. So look out for more videos coming soon. Take care, guys.